Hello, my name is Noah Martinez. I did mentorship one this summer at North Carolina School of Science and Math, and I went to North Carolina State University every day, and I worked on the split mNeonGreen fluorophore project towards a system to visualize proteins interacting in single cells. So, for some background, genetically encoded fluorescent proteins can be used to understand protein-protein interactions. And what this does, it allows us to track interactions dynamically over time and study how these interactions work within specific cells and under which conditions they interact with each other. The more specifically, the split fluorescent protein allows the visualization of these protein-protein interactions. And really, what happens is these fragments fluorescent only when brought together by interacting proteins. So my research question for this was, how can a protein protocol be optimized to construct plasmids containing two halves of a fluorophore? So one of the first objectives for this was to use different concentrations of ampicillin on an LB plate. So one time we tried 25 microliters and the other time we tried 15 microliters of this ampicillin to see if that would change how much colony growth we could get. Another option was to optimize the storage of the JM109 cells or J cells, which are E. coli cells that are required for these transformations. And the final objective was to see how different salt concentrations of transformations would affect. So for our methods, to get to these points where we can try these different protocols, we had to first do three main steps. The first one was the restriction digestion using pack and ascites, which cuts the DNA into a little pieces where we get the mNeonGreen 1-10, the mNeonGreen 32A11, and the London 26 backbone, and it's only a certain amount of that DNA being used. Once we did that, we got a gel electrophoresis and found the DNA that we needed to cut out of that gel, and then we spun it down for DNA and we nano dropped it. And we would get our DNA fragments would be anywhere from 3 nanograms per microliter to up to 20 nanograms per microliter, which is very small, but for how diluted it was very good numbers. And then once we did that, we had to ligate the purified DNA pieces so that we could just have the DNA and sort of just like add the mNeonGreen 1-10 to the London 266 and the mNeonGreen 32A11 to also the London 26 backbone. And once we did that, we could finally try this transformation into the JM109 cells. So the first thing we did was try different ampicillins and to see if either of those worked. And then we also tried the different salt concentrations, and we also tried having in-house cells, which we created on our own, versus company cells, which were sent into us. Our results were very lacking of growth of colonies, so we really couldn't tell. And with that, like our conclusion is that changes in these parameters did not influence the efficiency, so we couldn't really find an optimal protocol to use. And so really the implication of these projects was to find the optimal protocol to see fluorescent light in the split fluorescent plasmid. Future research to be done towards more protein-protein interactions with that using yeast cells. And then we also have some more future actions of using different split fluorophores to investigate the protein-protein interactions over time. Like I use the mNeonGreen, there's other fluorophores that will be used, and also to really explore how they interact with each other and how that works, and also looking at reversible protein-protein interactions within a cell. I would like to thank my NCSM mentors, Dr. Sarah Shoemaker and Dr. Zoe Lowe, as well as my North Carolina State University mentors, Dr. Orlando Arguello Miranda, my PI, and Bryn Merritt, my life technician, who did a fantastic job helping me this year. I'd also like to thank the Research and Innovation Program, the NCSM Foundation, the Burroughs Welcome Fund, and North Carolina State University College of Agricultural and Life Science for allowing me to use their lab this year.